So we've looked at what types of mutations exist. Now let's think a little more about mutations in a bigger picture. So here's how it happens. When this cell is trying to divide, it needs to copy its genome. So it needs to synthesize an entire new DNA molecule, and it does it by adding one base at a time. It compares it just looks for a base that fits or properly base pairs, and if it finds one, it adds it, and then it does it again and again and again, roughly 2 million times or 5 million times, depending on how big the bacterial chromosome is. That process can have errors. It's entirely possible for something to go wrong and incorporate the wrong nucleotide. So if that happens, one of the DNA molecules will have that error and the other one won't. So here's where the mutation happened. This daughter cell gets it, this one doesn't. So now these two daughter cells are genetically distinct from each other. This one has a mutation that this one doesn't have. And that can lead to lots of different situations. So this is a question you might want to ask. Are mutations common? Yes. Biologists think about mutations all the time. We see mutations all the time. There's a kind of a continuum of mutations, from silent mutations, or mutations that don't change anything noticeable, to lethal mutations that inactivate a critical gene. So we often detect these in the population because, if we go back, if this is a silent mutation, there's no real difference between those two cells, between these two cells. And so it's entirely possible that this one might just reproduce slightly faster than this one for other reasons, and this mutation will become part of this species. On the other hand, what if this was a lethal mutation? Well, what would happen next is this cell would divide, but this one wouldn't. And that's what would happen. So we would never see this in the population. Because this one would divide and you'd get 1 million great-great-great-great-granddaughter cells or something like that, and this would have 0. So mutations can be everything from not detectable to completely lethal. And it's rare that a mutation is going to really change the behavior of a cell in a way that helps the cell. If that happens, that mutant trait will become common in the population. So the example we're going to see later is antibiotic resistance. If you have a bacterial infection and Staphylococcus aureus is damaging your tissues and growing as fast as it can, if you then, let's just say for giggles, you inject a huge amount of vancomycin into your bloodstream, that's really poisonous to Staphylococcus aureus. Most Staphylococcus aureus will die pretty quickly if a large dose of vancomycin hits them. But maybe they have a mutation that makes them resistant to vancomycin. Maybe one of them just gets this random mutation. Well, you can imagine all of its buddies die, and it doesn't. It gets to grow as much as it wants. It gets to all the resources that its buddies were going to have. It gets all of them. So that's kind of what we're thinking about here. So how often does this happen? That's what we're thinking about next. We know mutations do happen, and we're going to look, we're going to go back to that antibiotic resistance example later, and if you didn't get it, don't worry about it. How often does this happen? That's important because, well, as you'll see, we can predict various things if we know the mutation rates. So what we can start with is the error rate of a polymerase. So for example, DNA polymerase complexes like we have, they have a certain error rate. They'll make rates, errors at between 10 to the minus 8 and 10 to the minus 10. So that means 1 out of every 10 to the 8th to 1 out of every 10 to the 10th. So that's potentially 1 error per every 10 billion nucleotides. So our DNA polymerase can be incredibly precise and accurate. Just imagine typing a message 10 billion letters long and only making 1 error. That's what our DNA polymerase can do. And I'm not sure what conditions drive it through this drastic change from 100 million to 10,000 million. But other types of organisms might have very different error rates, like RNA viruses that have to encode their own RNA-dependent RNA polymerases. Most of them don't have any proofreading ability. Most of them can't detect errors. So while part of this really great error rate, it comes from our ability to proofread. So part of our DNA polymerase has proofreading and correcting capabilities. Most RNA polymerases don't have proofreading, so their mutation rates are much higher, or their error rates are much higher. Whoops. We can do some math here, and we can compare, like, how big is your genome? How many base pairs? Well, it's about 3 billion base pairs long. And you make somewhere between 1 error between 100 million bases or 10 billion. If the error rate is like this, you're going to have several mutations every time your genome is copied. If this is your error rate, you're going to have, every daughter cell you make will have mutations. If this is the error rate, you'd have to divide about 3 times before you'd expect to see a mutation. So, okay, we can't make a precise prediction from these numbers because these numbers are big, broad estimates, but what we can say is we wouldn't be surprised if every cell in your body has 1 mutation. We wouldn't be surprised if every cell in your body has 10 mutations. We would be surprised if every cell had 1 million mutations. And that's the kind of analysis we're doing here. So we don't worry too much about those mutations in human cells because a lethal mutation in one of your epithelial cells will lead to that cell not successfully reproducing, but maybe it wasn't going to reproduce anyway. A lot of our cells don't divide. But in bacteria, mutations can rapidly make a difference. So this slide is where we do some of those calculations. Again, DNA polymerases can proofread, so they might make an error somewhere in the middle of that range I gave you before. So maybe 1 out of every 10 to the 9th bases. So if we have 1 error out of every 10 to the 9th bases, we say the error rate is 10 to the negative 9. So we just divide 1 by that number. And we can bring some other numbers into it. We can bring in the total number of bases in a bacterial genome, which is around 5 million, or 5 times 10 to the 6th. So what do we do with these numbers? Well, let's look at it. So our 2 numbers are, let's see, it was one error per 10 to the 9th bases. That's one of our numbers. And let's see, our other number would be like, 5 times 10 to the 6th bases per cell. So those are our 2 numbers. There's a limited number of things we can do with 2 numbers. So what I look at is the units. Oop, there went the screen. Well, anyway, I'll bring that back in a second, but what I want to see are, what are the units, and can I figure out how many cells I would have to look at before I found a mutation. So I'm looking for an answer with the units cells per error. And let's see if we can bring back the screen and figure out how to do that. So what we can do is we can flip these over, because as long as you also flip the units, it doesn't change anything. So I could just as easily say 10 to the 9th bases per error. And that still makes sense, because you have to look at 10 to the 9th bases to find one error. So it's 10 to the 9th bases per one error. And I could put this on the bottom. I could say one cell. I could actually just multiply this times this, and I'd flip this over too. So on top I'd have one cell per 5 times 10 to the 6th bases. One cell has that many bases in its genome. And again, as long as the units follow the numbers, we're okay. We can put this on the bottom, just make sure the units flip also. And so if we do this calculation, I'm sleepy, so I'm not going to try it, but I did it already. What we end up with is around 200, or we end up with 200 cells per error. That is, you have to look at 200 bacterial cells before you find an error. So if the genome is that many bases, and that's how many bases you have to look at to find an error, you have to look at 200 cells until you've looked at that many bases. So 200 times that gives you that. I'm not as interested in you being able to do that calculation as I am in you having a sense of how common these mutations are. So again, we came up with roughly 1 out of every 200 cells will have a mutation. So is that a big number? 1 out of every 200, it takes forever to count to 200, so how many do you see? Well, bacterial numbers are huge. So I'm just thinking, like, if I'm doing research in a lab, I start a culture before I go home for the day. I put some cells into some broth. When I come back the next day, they've grown, and I have lots of cells to work with. Do that in a test tube. It's a routine thing we do, call it an overnight culture. That would have roughly 5 times 10 to the 9th cells in it. And so we can go back and look at our math. We have 200 cells per error, and I have 5 times 10 to the 9th cells per culture. So what I can do is multiply. I can have some fun here. 5 times 10 to the 9th cells per culture times, we're going to flip that, so 1 error per 200 cells. And what do we get? I'm not going to do this when I'm this sleepy. What we get is 2.5 times 10 to the 7th mutants in a typical culture. How many is that? That's 25 million. So 25 million different mutants. What it basically boils down to is mutations are happening everywhere all the time. Within my test tube, if I want to find a cell that has a mutation in any particular gene, there's one in there. The numbers during a bacterial infection of a human could be much higher. So if a person has a really bad staphylococcal infection, they could have 10 times that many cells in them. And if one little mutation could make that cell resistant to an antibiotic, that mutation has probably happened, and that mutant is in there somewhere. So you have to assume that mutations are going to happen everywhere very rapidly. It gets even more intense when we look at things like viruses. So this is a comparison of different types of creatures and their mutation rates. So down here we see mutation rate. 10 to the minus 9 was the one we were looking at before. That'd be like prokaryotes. You see different numbers for humans for the error rate, but we could say that like multi-celled eukaryotes might have this really great error rate. So that's 1 error per 10 to the 10th bases, or 1 error per 10 to the 11th bases. And then here we're just looking at how big is the genome. How many bases does the cell have to put together to make a new cell? And it kind of makes sense that if you have a genome that's 10 to the 10th bases, you need a mutation rate that's very low. If you have a genome that's 10 to the 4th bases, like a lot of viruses, you don't need that amazingly good proofreading ability, because you can make a bunch of viruses without any mutants or without any mutations in them. A lot of your viruses will be fine. That's one thing we can take from this. Also, of course, this is a exponential scale, right? We're going 100, 1,000, 10,000, 100,000, a million, 10 million, 100 million, billion, 10 billion, 100 billion, right? What I want you to look at is different types of things we've looked at. Lower eukaryotes would be like fungi. Prokaryotes are bacteria and archaea. Double-stranded DNA viruses. We will see some later, but like smallpox virus. Single-stranded DNA we haven't really looked at. And then RNA viruses would be HIV and influenza and SARS-CoV-2. These have RNA genomes. They have the highest mutation rate. That is, you might be able to find in influenza 1 error in every 10,000 bases. Whereas for human, you might have to look at 10 billion bases to find 1 error. So this mutation rate tells us how fast these things are going to evolve. It really does matter. So one is that bacteria will mutate faster than we will, but also they reproduce faster than we do. So their populations evolve. We have to be ready for that. DNA viruses evolve slowly enough that we can get good vaccines against them. So smallpox is a good example. We eradicated it because we had one vaccine that worked all over the world until smallpox disappeared. And that's pretty awesome, because smallpox was one of the worst things that ever happened to humanity, and it's gone. Or now it only lives in laboratories in the US and Russia, to our knowledge. RNA viruses are different. Like influenza, in about 7 years, any given influenza strain will evolve enough that our immune systems will forget it. And it'll be different enough from the vaccine we developed for it that we need a new vaccine for it. So that's a number I read in an article where they were talking to vaccine designers who were comparing influenza to SARS-CoV-2. And they said, yeah, it takes about 7 years before we need to make a new vaccine for a given influenza strain. SARS-CoV-2 and other coronaviruses are actually unusual in that they proofread their RNA as they're making it. And so they have lower error rates than something like influenza. So why are we so worried that we have to keep making different COVID vaccines? Why are we worried about variants? Well, there are huge numbers of people with COVID infections. As I'm making this video, there have been 100 million people around the world who've been infected with SARS-CoV-2. That is an astronomically huge number of virus particles that have been made. And every time a virus particle gets made, there's a chance that it could be a mutant. And so when you have that many people infected by the same virus, you're going to get really fast production of mutants and really fast evolution of mutants as they try to evade our immune systems. So even though SARS-CoV-2 has proofreading ability, since it's replicating in so many people, that doesn't matter. Right. And what's even more amazing than SARS-CoV-2 is HIV. HIV is an RNA virus. Well, it's a retrovirus, but RNA that mutates so quickly it can evade our immune system just within one person. So HIV mutates so fast that we can't make an old-fashioned vaccine for it. We can't make the kind of vaccine that we would use for polio or smallpox or even SARS-CoV-2. So influenza evolves fast enough that we need a new flu shot every year. And if epidemiologists could give us 2 or 3 flu shots every year, they probably would. But HIV mutates even faster. And you might say that different species will appear within a single person who has HIV. So I'm going to show you 3 phylogenetic trees that are all to scale. So they're all on the same scale. These are the 3 trees. This is an old study, and this is one of the most amazing diagrams I've ever seen. This is a phylogenetic tree of all influenza isolates in the world in 1997. And this is a phylogenetic tree at the same time of all the HIV isolates inside one person. So everything in the world for influenza, everything in one person for HIV. And this is a phylogenetic tree of all the HIV in one country. So if you measure the number of differences between this and this, those viruses barely resemble each other, and they'd have a few commonalities that make them both HIV, but there's no way a person's immune system, having seen this, could protect you against this. And smallpox is on the other end of that scale. It's a DNA virus, and so its evolution is slow enough that we can protect ourselves from it. Okay, so that's mutations, and that's error rates and stuff like that. And again, what I want you to know is, if anything, the conclusions we came to, like how many bacterial cells are mutants. Is it every cell? Is it 1 cell out of every million cells? No, it's something like 1 out of every 200, roughly. Yeah, so in the next video, we will look at ways that microorganisms share genes on a bigger scale than just having mutations. So I'll see you there.